Hi, everybody. We're going to be presenting on Module 3, Human Microsatellite DNA. So there's been this buzz all around campus about ancestry tests, but I can't join in because I know nothing about my ancestry.
Hey, you there. Have you ever wondered how you can learn more about your ancestry?
Yeah.
Are you curious about some of the biology behind DNA?
Yeah.
Do you want to spit in a cup?
Yeah. Wait, what?
Welcome to DNA Today. You spit in a cup and we'll hook you up.
That sounds interesting. So how does this process work?
Well, I'm glad you asked. First off, do you know anything about DNA?
Well, sure. We learned about it in my bio class. DNA stands for deoxyribonucleic acid. It's a double helix made up of a negatively charged sugar phosphate backbone and 4 nucleotides: adenine, guanine, cytosine, and thymine.
Very good. Did you know that microsatellite DNA are distinctive bands of repetitive sequences, usually 1 to 6 base pairs long, that are separate from other non-repeat or single-copy DNA? In the forensic community, microsatellites are known as short tandem repeats and are used for human identification.
That's cool and all, but I thought I was just spitting in a cup. What does all of this information have to do with learning more about my ancestry?
Well, this has everything to do with ancestry tests. How about we turn this over to one of our trusted customers to describe the process?
Hello there. Oh my gosh, can I just say that I'm so excited to be here with DNA today? Their ancestry test changed my life. I feel like a whole new person.
Get to the point.
Oh, right. Sorry about that. So one of the most important parts of this process is isolating your DNA.
So how do you do that?
Oh, it's simple. After you spit in a cup and shake up your DNA a tad, you just got to use some Lysis Buffer and it'll break down the cell and nuclear membranes.
So the Lysis Buffer contains chemicals able to disrupt cell membranes and break down proteins associated with DNA in the chromosomes. It also denatures enzymes that can cleave DNA. The solution is vortex, and DNA is released from cheek cells and binds to the surface of the resin beads. The tube is then placed on the magnetic stand so that the resin beads congregate together. The Lysis Buffer is then pipetted out, leaving only the resin beads with DNA attached to them in the tube.
Wait, breaking down stuff? Won't that, contaminate my DNA or something? Are you sure about this?
Actually, quite the opposite. This is one of the steps to help you decontaminate your DNA.
Decontaminate your DNA? Like giving your DNA a bath or something?
Exactly. Well, maybe not exactly, but you get the point. You'll be using a wash buffer to remove the residues from the DNA.
So the wash buffer. This contains salt and ethanol to remove organic components such as lipids and protein from DNA or RNA molecules while still leaving the DNA molecules attached to the beads. 100 microliters of wash buffer is transferred into the tube containing resin beads, and the sample is vortex, then placed on the magnetic stand. The wash buffer is then pipetted out, and the process is repeated another 2 times.
Okay, so now that I've somehow broken down my DNA in cell membranes without hurting it and given my DNA a bath, now what?
Yeah, isn't this exciting? Next, we're going to use some handy-dandy elution buffer to separate the DNA from the magnetic bead. And congrats, now you're finally done preparing your DNA.
Wait, preparing? You mean we're not done?
100 microliters of elution buffer is added to the tube, and it is vortex for 2 seconds. It is immediately put in a 65°C bath for 5 minutes. The tube is removed from the water bath and vortex for another 2 seconds and immediately placed onto the magnetic stand. The elution buffer works to extract the DNA from the resin beads so that the solution now contains the DNA. Once this process is complete, the solution can be placed into the wells of a thermal cycler for PCR process. The solution is extracted from the beads with the micropipette, and the foil of the well is punctured using the tip of this pipette. Once the tip is inserted, the purified DNA solution can be released into the well.
Of course we're not done, silly. Do you really think one strand of DNA is going to be enough? We got to replicate it until you have a gazillion strands.
What? Am I supposed to be copying and pasting my DNA or something? How on earth can I make 1,000,000 copies from one?
It's easy. You just got to use the polymerase chain reaction, you know?
No, I don't know. Nerd.
Okay, using PCR, you can exponentially amplify a segment of DNA to make thousands to millions of more copies.
So the polymerase chain reaction, or PCR, is a technique to make many copies of a specific DNA region in vitro, essentially in a test tube rather than an organism. PCR relies on the thermostable DNA polymerase, Taq polymerase, and requires DNA primers designed specifically for the DNA region of interest. PCR primers are short pieces of single-stranded DNA, usually around 17 to 28 nucleotides in length. 2 primers are used in each PCR reaction, and they are designed so that they flank a particular target region. Their base composition should be 40 to 60% of guanine cytosine pairs, and they should also have similar melting temperatures between primer sets, usually between 55 and 65 degrees Celsius. Choosing the correct primer is key to the process, as they must only amplify to a single location in the entire DNA sample.
I guess. So, are we done now?
Here's the fun part. Now you have 2 ways to analyze your DNA. Do you remember that DNA into a well way back when?
Yeah, what about it?
Well, the first method uses the electropherogram, which is this really cool plot that can tell you specific genotypes based on the length of DNA fragments.
So an electropherogram is a chromatogram that with the STR results that one can use to calculate the probability of finding another individual with the same genotypic profile as yours. It is essentially a multicolored graphic result of an extremely fine-tuned electrophoresis. The color peaks represent the color of the fluorescent labels on the primers that indicate possible loci. STR loci have been used as genetic markers for the purpose of human identification because of their broad range of variation and the observation that they do not outwardly express any phenotype. In the U.S., 13 of these loci are used for forensic identification.
Okay, so what's the dealio with this second method?
Oh, it's this thing called gel electro.
Oh, bro, hold up. Gel electrophoresis? I learned about this in AP Bio. This is that thing where DNA moves from the positive end to the negative end because of its phosphate group. It makes it negative.
Yeah,
And then you can measure how far the DNA segments traveled, with the smaller segments going farther than the larger ones.
now you're getting it.
So gel electrophoresis is basically just allows you to visualize the DNA migration in real time. You basically already hit all the key points. In a gel electrophoresis, an electrical field is established in a medium through which charged particles can diffuse. The negatively charged phosphate ions in the nucleic acids cause the nucleic acids to migrate to the positive pole of the field. Smaller molecules will move more rapidly through the gel electrophoresis than larger molecules and end up closer to the positive end of the gel. Gel electrophoresis basically just separates the DNA fragments by size.
Okay, but how am I supposed to remember all of this?
We've got just the thing for you. If you order by the end of this lecture, you'll receive your very own DNA Today mixtape. I can assure you, it's fire.
Are you sure about that?
Positive. Why don't we give you a little sample? You can sing along if you'd like.
I walked in my 1500 lab, we were looking at our DNA. They said, y'all are about to feel so fab because we're spitting in some cups today. First we'll take 100 microliters of the lysis. If you don't use it, then there's gonna be a crisis. Then watching the magnetic bead moves so the DNA is freed. Finally we'll use the buffer evolution. Make sure to check for no confusion. Don't forget to vortex it, then you've got the hot water bath. We're gonna put it in the water bath. It's going in the water bath. Then I take my tube out of my pipette. We're extracting our DNA. Some polymerase chain reaction. We're gonna put the DNA in action. Let's look at electropherogram. I can post a pic to Instagram. Yeah, 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 yeah. It's a party with my DNA. Yeah, 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 yeah. It's a party with my DNA.